Can everybody hear me? Hi, my name is Maiya Duncan, and this is my dissertation research on genetic and behavioral divergence, a comparative study of hybrid and wild mallards in coastal South Carolina. So just some background on mallards and basically how they got here and their stalking history. Mallards are vital for hunting and food. They originally nested west of the Mississippi River and expanded eastward by the 1950s. Their introduction to Eastern North America occurred through breeding range expansion and game farm releases, starting in the 1920s with their expansion driven by natural migration and intentional game farm releases from the 1920s to the 1960s. This large scale introduction has played a crucial role in shaping the current distribution and population dynamics of mallards across these regions. The genetic decline has in wild mallards is caused by hybridization with domestic mallards, and that has significantly altered the genetic structure, the genetic structure of wild mallards. And basically, with these releases, there have been over 500,000 releases of these mallards in the United States. And the consequences basically has came from these genetic changes. And these genetic changes have caused these birds to have a heightened predation risk, and that has caused posed a long-term survival challenges. And the picture I have on here is from Delta Waterfowl, and it basically shows the range of these mallards and where they breed, specifically in the prairie pothole region. And the red dots represent those juvenile mallards that were banded in the prairie pothole region and where hunters harvested those mallards. So as you can see, they are migrating across the United States, but these typically are wild mallards, which do have that migration behavior in them. So why is this research important? Why should we care that mallard genetics are endangered by these game farm releases? So the first is being genetic consequences of stalking. Discussing the genetic consequences has led to game farm mallards disturbing wild mallard genetics. These birds trace their lineage back to Eurasian domestic stock, and they've introduced non-native genes into wild populations in both North America and Europe. Research by Dr. Phil Aretsky shows that this gene flow has compromised the genetic integrity of wild mallards. In other words, these releases aren't just increasing population numbers, they're altering the fundamental genetic identity of wild birds. And over time, hybridization and gene flow have significantly reshaped the gene pool across regions. And this isn't an isolated phenomenon, as it's been documented with similar genetic consequences across both continents. And the picture I have on the screen is showing all the different breeds that have resulted from the domestication of wild mallards. And as you can see, a game farm mallard is very similar to a wild mallard, but their behavior is very different. As far as game farm mallards are very habitualized and used to human presence, they will not have that fight or flight response when you do approach them. They actually will allow you to handle them, compared to a wild mallard. As we know, wild ducks will fly away from you if you do approach them. So this project is composed of three different objectives. Objective one is focusing on gathering essential data regarding the movement patterns, survival rates, reproductive success, and genetic makeup of game farm mallards. And I aim to understand how these domesticated birds differ from their wild counterparts, particularly in terms of their behavior and adaptability to the environment. And for my first hypothesis, I believe game farm mallards will exhibit reduced movement ranges and lower survival rates compared to wild mallards, indicating potential genetic limitations and habitat changes. For objective two, it involves evaluating the same parameters measured in objective one, but focusing on the newly integrated population of the newly integrated populations of game farm mallards. And with this integrated population, I believe it will show once they are around wild mallards, their behavior might mimic them, and they might actually start to show the migration behavior. And for objective three is to analyze the predator-prey dynamics with the wild, hybrid, and game farm mallards, and basically to see, okay, we have already looked at their behavior. Do they have that fight or flight response when it comes to predators in the area, or will they just stay and allow themselves to be predated on? So the methods for this project are broken down into four different sections. The first is assessing genetic and ecological impacts. So our primary goal is to assess how the release of game farm mallards, particularly those with European ancestry, may be affecting wild mallard populations in South Carolina. These birds are raised for hunting purposes, but often lack the behavioral and genetic traits that allow for successful integration into wild populations. We're especially concerned about the potential hybridization, which could reduce the genetic integrity of wild mallards, as well as behavioral changes that affect things like predator avoidance, migration, and reproduction. So to evaluate these effects, we'll be conducting genetic sampling and comparing survival nesting success and behavioral differences across wild, hybrid, and game farm individuals. And the predator-prey dynamics will also be assessed using field observations and camera traps. So for the second part, using telemetry and monitoring, we will be banding 250 game farm mallards and tagging them with VHF GPS and tracking devices to see where are they going, are they surviving, are they actually leaving this area, are they staying in the impoundments that they were released in. And as you can see the pictures on the screen, I have the VHF GPS tracker that we will be putting on the mallards, and also the MODIS towers that we'll be using as well. The VHF trackers will be for close up tracking, like actually going out in the field, monitoring and looking for them. And the MODIS towers will be more so for long distance tracking to see, are these birds being pinged by different MODIS tower sites across the state? And are they actually leaving this area? And are they actually being pinged at other MODIS towers locations in other states as well? The next one is trapping and sampling protocols. So eventually we will be using a swim-in funnel trap, as you can see, and we will be trying to see if we find the birds that we banded initially in the beginning and recapture those and seeing, okay, have they gone anywhere? Are they still local? And if they are local, we know for a fact, okay, these probably are game farm mallards and they are not moving around in the state. And we also want to take DNA samples from those birds as well, including the 250 birds that we had initially banded in the beginning. And we will give those to Dr. Filaretsky and actually find out what is the genetic integrity of these birds and find out how many of these birds in the state are actually game farm mallards, are some of these birds that we're finding wild mallards. And lastly, for management implications, we want to be able to compare the movement, survival, and fitness of game farm mallards versus wild mallards and see if that can spark some type of conversation about how can we in the future try to preserve these wild mallards and preserve their genetic integrity. And the last picture on the screen is a DNA sample that we will be using to collect from mallards. And it's just a little bit of blood just to see what is their genetic integrity. So the project so far has been done in phase 1, and eventually we will go into phase 2. So phase 1 is banding of juveniles and collecting DNA samples. So our primary goal is to better understand the movement patterns of juvenile game farm mallards once they're released into the landscape. And these birds are banded and sampled, which means we collect tissue or blood samples for genetic analysis and assign each bird a uniquely coded leg band. This way, if they are later recovered, either through hunting or observation, we can match their band number to their DNA and other relevant data. So far, 85 juvenile game farm mallards have been banded and released across several private properties in coastal South Carolina. And these birds were released at a young age. So we know for a fact that their genetics are game farm mallards and there is no wild genetic influence. The main goal of this phase isn't to uncover genetic variation, but to track where these birds go post-release. And do they remain in the local area or do they disperse to other parts of the states? And for phase 2, that will actually be putting the trackers on adult birds as they are big enough to handle the weight of the tracker. And we're basically trying to see when these game farm mallards are released during the winter, with the migratory birds present, are they mimicking their behavior and actually migrating with them? Or do they stay in this area and basically conform to staying as game farm mallards? And with this, we'll be doing 100 adults. 50 of them will be getting the MODIS nanotags that will be pinged by the MODIS towers, and 50 will be getting the VHF trackers. And the pictures on the screen are the leg bands that we will be putting on the birds and the vials used to collect their DNA sample. So my anticipated results for this project, based, based on previous research, we anticipate that game farm mallards will show reduced ecological fitness in the wild. Specifically, they may have lower movement ranges, reduced survival rates, and be more vulnerable to predation. And this is largely due to behavioral deficits, such as poor predator recognition and a lack of adaptation to natural wetland environments. For the post-release movement, one of the main goals of this project is to understand what happens to these mallards after they're released. Specifically, what is their movement patterns? So far, we've been focusing on juveniles in the phase 1. And the key question is, do these birds stay close to where they were released, or do they disperse more widely, possibly even across state lines? And lastly, for ecological interaction, we want to see when hunting season comes and winter time comes, we want to evaluate if game farm mallards will join the wild flocks and migrate, or will they just stay isolated in their areas? I'd like to thank my collaborators, South Carolina Waterfowl Association. They've done so much work on this project this summer with banding the 85 juvenile birds. I'd like to thank Dr. Loretsky and his lab at University of Texas, El Paso, Mark Johnson and his organization, Paul Maddow Waterfowl Conservation, and the James C. Kennedy Center. Any questions? Anyone have questions? Okay, thank you.